Hi everyone, welcome. My name is Akanksha Nagarkar. I'm an undergraduate researcher at the Downs Lab at UMass Amherst, and today I will be presenting my research, which is titled The Generation and Characterization of a Null GABAA Receptor Alpha-4 Subunit Danio rerio line. Let's get into it. So first of all, let's understand what GABA is. GABA stands for gamma-aminobutyric acid, and it is the chief inhibitory neurotransmitter of the mammalian central nervous system. Here we see a structure of what it looks like. And because it is an inhibitory molecule, it's involved in numerous neuropathologies like epilepsy and Parkinson's disease. And GABA carries out its work by binding to a GABA receptor. For my research, I focused on the GABAA receptor. So what is the GABAA receptor? The GABAA receptor is an ion channel that is embedded in the membrane of a neuronal cell, as we can see here. It is selectively permeable to chloride ions. So when a neurotransmitter like GABA binds to the receptor changes shape, and chloride ions are allowed to flow through the receptor's ion channel. Under physiological conditions, the chloride ions flow into the neuron, and this lowers the chance of an action potential occurring. So in this way, the receptor has an inhibitory effect on neurotransmission. The structure of the receptor itself is pretty interesting as well. It's made of multiple subunits, as we see here, and we know now that there are 19 possible ones. Although we get lots of combinations of subunits, the alpha subunit, this one right here, is consistently present at the GABA binding sites, which is pretty significant. Another interesting feature of the GABAA receptor is that it's highly conserved across vertebrate animal species. In the lab, we use the zebrafish as a model organism to study GABAA. And zebrafish are an excellent model organism for many reasons. First of all, they develop outside of the mother, and they develop transparently, which we can see here. They also develop very rapidly, and at about 5 days post-fertilization, neural systems involving sensory and motor networks are established. So because of this, the way that they develop, we're able to access the nervous system throughout all the stages of development. They also have a pretty large clutch size, which gives us a lot to work with. Their genome is fully sequenced, and they have the same major organs and tissues as humans. So that makes the zebrafish an excellent model for human disease as well, like epilepsy. So what does GABA signaling look like in zebrafish? Previous studies have shown that interfering with GABAergic signaling leads to hyperactive behavior in the fish. This makes sense because GABAergic signaling often leads to inhibitory responses. If we interfere with normal GABAergic signaling, we might expect to get hyperactive behavior. Also, previous research in the Downs lab found that there are 8 GABAA alpha subunit orthologs in the zebrafish, and these are here. My research focuses on the alpha-4 subunit in zebrafish. This is because in previous research efforts in the Downs lab, we found that knocking down the alpha-4 subunit while also simultaneously knocking down other subunits showed a hyperactive phenotype in the fish. Furthermore, the expression of alpha-4 was seen to be in discrete cells throughout the brain, including in the hindbrain. Now, this is a very important part of the brain because it's involved in essential functions like breathing, as well as muscle control in certain areas of the body. Despite how significant alpha-4 seems, its phenotypic expression has not yet been characterized. Therefore, the first objective of my research was to create a stable line of zebrafish in which alpha-4 expression was knocked down in all cells. The next step of my research was to analyze the phenotypic effect of this knockdown and essentially characterize what alpha-4 may be responsible for. I first needed to use CRISPR-Cas9 to knock down the gene responsible for alpha-4 expression, This gene is called GABAA4. The resulting fish would be mosaic because CRISPR-Cas9 induces a lot of different mutations, and these mutations would be in some of the somatic cells of the fish. They wouldn't be germline, and so not all of the cells would carry this alpha-4 knockdown mutation. And I wouldn't really be able to make any concrete conclusions about alpha-4's phenotypic expression. In order to do that, I would need to ensure that the fish had alpha-4 knockdown in all of their cells due to the same mutation. In order to do this, I needed to perform a series of crosses with the mosaic fish until I got a generation of fish that were homozygous for the alpha-4 knockdown mutation. Then I could go ahead and do analysis on the behavior and phenotype. So here's a schematic of the process that I used. First, I used CRISPR-Cas9 to target the GABAA4 gene and create mutations. Then I genotyped the resulting fish to identify what mutations I had created. Then I performed a series of crosses and genotyped them until I ended up with an F2 generation in which there were fish that were homozygous for the GABAA4 knockdown mutation. And then I did phenotypic and behavioral analysis. So a significant tool in this process was CRISPR-Cas9 because I used CRISPR-Cas9 to target the GABAA4 gene and create mutations such that the expression of GABAA4 was knocked down. In order to do this, I first designed guide RNA primers targeted to GABAA4, and then I injected this guide RNA and Cas9 enzyme cocktail into wild-type embryos in the single-cell stage. This caused mutations that interfered with the gene. I was then able to go ahead and identify which mutations I had created. So one of the mutations I had created was an 11 base pair deletion in exon 4 of the GABAA4 gene. After the series of crosses and genotyping, this is the mutation that the resulting line of alpha-4 knockdown fish all have. And this is kind of a schematic of what it looks like. So once I had the stable zebrafish line that were homozygous for this alpha-4 knockdown mutation, the next part of my research was to analyze the behavior of the fish. One of the assays I did was touch-evoked behavior, and this involves tapping the head of a zebrafish at various developmental stages with a probe, and then analyzing any startle responses that followed. A startle response, also known as escape behavior, is when the zebrafish bends its body in a C shape before swimming away rapidly from the stimulus. This is what it looks like in wild-type fish. So you can see that we tapped it on the head, and then it swims away. And now let's look at what a mutant would look like. You can see that it's bending and it's doing a lot of C bends. Pretty abnormal looking. It's a pretty good indication, these sort of C bends, of locomotive behavior and a disruption in normal GABAergic signaling. So this whole process is done in a chamber with a high-speed video camera to film the responses. I then used a larval zebrafish tracking program called Marigold, which was developed in the Downs lab to analyze the videos and then determine fish movement and acceleration at various time points. So I analyzed the videos using Marigold, and I looked at the number of C bends as well as the swim duration in response to the probe. And I looked at fish that had one normal copy of the alpha-4 protein, as well as fish that had alpha-4 completely knocked down. And these are essentially this is the null line that we're looking at. And we don't see any statistical differences between the two, and I used a t-test to do this. However, the N was pretty low, and this is all ongoing data analysis. This was only looking at 9 fish. And so this was probably responsible for high variability among samples and no statistically significant differences. But it'd be interesting to see what we get going forward. So another assay that I did was light flash. And this involves placing zebrafish at various developmental stages into a chamber with a high-speed video camera as well as a light source. The fish are first acclimated to darkness for 30 minutes, and then they are exposed to a light stimulus. So this is what it looks like. It's dark and then a light stimulus. And from this, I'm able to look at the number of C-bends that the fish perform in response to the stimulus. And I also do the reverse. So I acclimate them to light for 30 minutes and then expose them to a dark stimulus to see what's going on. And the data analysis for this is still all ongoing, so it'll be very interesting to look at what's going on in terms of number of C-bends and overall behavior. From this research, I have created a null alpha-4 line that can be used in future experiments. Particularly, in a previous screen, I mentioned this a little bit before, we found that knocking down alpha-4 with other subunits reveals hyperactivity, and so this line can be used to investigate that further. Also, when I finished analyzing the data from the behavioral assays, I can make concrete conclusions about the role of alpha-4. In the grand scheme, this research provides insight into the role the alpha-4 subunit plays in GABAergic signaling. And that's it so far. Thank you all so much for listening, and this was definitely a team effort, and so I must acknowledge everyone in the Downs lab who helped make this possible. Thank you so much.